Not all artificial sweeteners are bad for you, and this one might actually fight cancer. You've probably heard of stevia, right? That plant-based sweetener used in everything from soda to protein bars? Well, it might be more than just a sugar substitute. A team of scientists has discovered that when stevia is fermented using a specific bacterium found on banana leaves, it transforms into something far more powerful, a potential cancer-fighting agent. Let's talk about pancreatic cancer for a moment. It's one of the deadliest out there. It spreads fast, resists most treatments, and has a 5-year survival rate of under 10%. Pretty grim. That's why researchers at Hiroshima University have been looking for better, safer ways to treat it, especially using natural compounds. So, they turned to a banana leaf bacterium called Lactobacillus plantarum SN13T and used it to ferment stevia leaves. They compared the fermented extract, called FSLE, to regular stevia, and the results were impressive. Now, this wasn't just tossing ingredients into a blender. The team carefully fine-tuned every part of the fermentation process: time, temperature, oxygen levels, you name it. The sweet spot? 72 hours, no oxygen, at 37 degrees Celsius. What they got was a powerful result. FSLE attacked pancreatic cancer cells significantly better than unfermented stevia, while leaving healthy kidney cells nearly untouched. That kind of selectivity is rare and incredibly promising for treatment. Instead of killing all cells blindly, FSLE weakened the cancer, slowed its growth, and even made the cells lose their shape and stop sticking together. Under the microscope, those cancer cells looked like a mess, but the healthy ones? Still standing. Even better, FSLE was also a strong antioxidant. It neutralized harmful free radicals with up to 94% scavenging power in one test and 75% in another. And when healthy cells were exposed to oxidative stress, FSLE helped them survive. So what made it so effective? It turns out fermentation created a new compound not found in raw stevia. It's called chlorogenic acid methyl ester, or CAME, a supercharged version of a common antioxidant. CAME was way more potent than the original. It stopped cancer cells from multiplying and triggered apoptosis, which is just a fancy word for making them self-destruct. Its IC50, the dose needed to kill half the cancer cells, was 119.1 g/ml, compared to 189.6 g/ml for regular chlorogenic acid. That's a big difference. CAME worked by locking cancer cells in place before they could divide, and then triggering enzymes that break them down from the inside. It even altered gene activity, boosting the ones that tell cells to die and suppressing the ones that help cancer survive. Basically, CAME wasn't just attacking cancer randomly, it was telling it to shut down in a very controlled way, through what's called mitochondrial apoptosis. And it all started with stevia, a simple, natural sweetener, turned into a precision tool against one of the world's deadliest cancers, all through fermentation with a single bacterium. The team believes this transformation is thanks to specific enzymes produced by the SN13T bacteria. Their next step is to test FSLE in live animals, starting with mice, to see how it behaves in real bodies with real immune systems. If the results hold up, this could open doors not just for fermented stevia, but for using probiotics and natural plant extracts as targeted therapies. Not bad for something that started out as a sweetener in your tea.